Beginning with the name of Allah, the most beneficent, the most merciful, I am Hafiz Muhammad Hammad, and I welcome you all to this tutorial on virtual screening using PyRx. So what is PyRx? PyRx is an open source software tool designed for virtual screening of molecular docking studies. It provides a user-friendly interface for molecular docking, structure-based drug design, and various computational chemistry tasks. So PyRx is built on top of popular molecular modeling tools, including Autodoc Vina for molecular docking and MGL tools for pre and post processing tasks. It allows users to perform virtual screening experiments by docking small molecules into a target protein structure to predict binding affinities and potential interactions. The software includes features for preparation of ligands and proteins. It runs molecular docking simulations and it also helps in analyzing the results. PyRx is often used in drug discovery and computational biology research to identify potential drug candidates and understand the binding mechanisms between ligands and target proteins. So next, as the name suggests, Py, it is a Python-based prescription. So what is Python? Python is actually a high-level, versatile, and general-purpose computer programming language. It was created in 1991 and it emphasizes on readability and simplicity, making it a popular choice for beginners as well as experienced developers. Because it is easy to learn and read, it is an interpreted language, it is a versatile language, it has a large standard library, it also provides community support, it is platform independent. So this aids in its object orientation and dynamic typing. Next, what is virtual screening? So virtual screening is actually a computational technique employed in drug discovery to predict the binding affinity of small molecules to a target protein, aiding in the identification of potential drug candidates. So this approach significantly accelerates the drug discovery process compared to traditional experimental methods. Virtual screening operates by simulating the interaction between molecules, predicting their ability to bind to a target. Its efficiency lies in the substantial time and cost savings it offers, making it an invaluable tool in drug discovery. It is also known as a repurposition study because of its powerful and valuable approach in drug discovery to find potential drug candidates for a protein or disorder. So the important steps in virtual screening include target identification and preparation. In this step, we retrieve the 3D structure of target protein from databases like PDB and prepare it for docking. Then we have ligand database preparation. In this step, we assemble a complete ligand database for our virtual screening process to ensure that structures are in appropriate format, which can be SDF or PDBQT, which we'll learn later. Then we perform ligand and protein pre-processing using tools like Autodoc tools and MGL tools or PyRx. Then we perform grid generation. Grid generation is actually an important step in which we specify the region where ligands will be docked using tools like AutoGrid or different other tools. Next is molecular docking, in which we allow the ligand molecule to dock or bind with the receptor molecule. Then docking pose analysis. Docking pose is actually a conformation or orientation in which ligand is attached or is bound with the receptor molecule. Next, pose selection and ranking. Poses are selected according to their binding affinities and the most promising candidates are selected. Then lead optimization is the iteratively refining of chemical structures and assessing the impact on binding affinity. There's interaction realization. This is done by using different tools like LegPlus, PyMol to realize the interaction of ligand with the residues of receptor molecule. Then we have tools for virtual screening. The most common tools used for virtual screening are Autodoc Vina, MGL tools, and terminal access, which is required to run Vina efficiently. And then we have OpenBabel, then OpenBabel for the conversion of STF formats to PDBQT formats. And then we have BioVia Discovery Studio, PyMol or LegPlus. Important terms in visual screening include SDF, which stands for Structure Data File, and it is a file format used for representation of chemical structures and associated data. It is commonly used in chemoinformatics and molecular modeling to store information about chemical compounds, including their 2D or 3D structures, properties, or annotations. Next, we have PDB. PDB stands for Protein Data Bank. It is a database and is also a file format for three-dimensional structure data of large biological molecules such as proteins and nucleic acids. It provides a standardized way to store and share structural information, enabling researchers to access and analyze the structures of biological macromolecules. Next, we have PDB-QT, which is an extension of PDB format, including information about charges and atom types for molecular docking studies. Because this accurate information is needed to visualize or simulate the ligand binding with the receptor molecule. Next, docking pose is actually a specific conformation or orientation of a ligand within the binding site of a target protein as predicted by a molecular docking simulation studies. Next, we have CSV format. CSV is actually a comma separated value format. It is a plain text file format where each line represents a row of data and values are separated by commas. Any data analysis visualizer such as Excel, Libre, Calc can open CSV and you can easily visualize CSV format files in there. Next, we have docking pose ranking on the basis of affinity. Affinity is the binding energy at which the ligand or pose is made, and ranking is the arrangement of binding poses according to their binding affinities. Next, RMSD stands for root mean square deviation, which is actually the average distance between corresponding atoms in two sets of coordinates, often used to quantify the structural similarity between two molecular structures. So, in docking pauses, we predict a dock with experimental or reference structure, providing a measure of accuracy of docking simulation can be analyzed with the RMSD value. Next, we have some links for PyRx. Mayavi is a visualizer. PyMol is also a visualizer. MGL tools for pre and post processing of protein molecules, and OpenBabel for the conversion of different file formats. Then we have PubChem database for our ligands, and Campbell database or Chembl database also we have for molecules or small molecules database. And then our CSP is PDB database, protein data bank, where we can download our PDB files. So let's jump into hands-on training for using PyRx. Okay, the first step in our PyRx tutorial would be to download and install PyRx in your system. And for this, open your search browser and type in PyRx. Hit enter and click on the first link. Here you can see Python prescription offers different options, but for now we'll go with the free option. For this, click on choose button, and you will be redirected to SourceForge page and hit download. This will automatically recognize your system as Windows, Mac or Linux and will download the relevant installation file. For now I am working on Windows machine, so this will be downloading exe file. Once the setup file is installed, open it and go with the prompts to install PyRx in your system. Let's open PyRx. For this, type in PyRx in your Windows Start menu, and PyRx Virtual Machining Tool, Screening Tool, and PyRx Virtual Screening Tool will open. Here you can see we have different wizards for aiding virtual screening, such as Veena, Autodog, OpenBabel, and Python 2.6 shell. To begin with, we have to download our protein molecules and structures first. So for this, let's move on to our search browser and type in PDB and hit enter. Choose this option and you'll be redirected to PDB databank page. Here you can search more than 200,000 of protein structures. For this tutorial, we'll be going with histone H3 protein that is related to DIPG, glioma disease. there is no PDB structure, experimental structure of histone H3 in the database. And if you can't find your structure in PDB database, you can also search for AlphaFold database. This is a complete database of computationally modeled structures. Let's type in histone H3, enter. And for Homo sapiens, and this will be our structure. Open it and we'll be downloading PDB file of it. File is downloaded. Next, we have to download our ligands. For that, we'll be visiting PubChem database. And the 3 FDA approved drugs for TIPG are ditriazoic acid, ganciclovir, and iodamide. So just type in here and we'll be opening their structures. On the ligand page, you can see the download button. Click on download and we have to download the 3D conformer SDF file, structure data file. Click on save. Same goes with the other ligands. Download SDF. Once our files are downloaded, let's move on to PyRx. In PyRx, you can import your molecule by clicking on File, Load Molecule, and choose your molecule here. Our molecule is AFA8. So our molecule is downloaded. You can realize it here by right clicking display options. You can also choose display option as molecular surface, and this is our molecule, protein histone H3 A chain. Okay, so for now we'll be working with this, and if you want to change or edit the chain, just click on this plus button and you can see all the residues. If there is any heat atom or any other chains you do not want in the structure, just right click on them and you can select it. By clicking on edit, you can remove them again. For now we are not going to do anything with that just structure. Now just right click on the structure and you can see an option of Autodock. Click on make macromolecule option and you can see your molecule will be created to a macromolecule. By moving to Autodock wizard, here is your molecule. Just visit Open Babel wizard now and you have to hit this plus icon one by one. There is only one limitation to PyRx, and that is you have to load each ligand one by one. Here we can, by clicking on it, we will import our first ligand, and now our second ligand, and same goes with the third ligand. After all the ligands are imported, you can see there is a molecular weight of each ligand listed here, and then the number of atoms it contains, and also the molecular formula. By right clicking on any of the ligands, just hit the option minimize all, and you will see all ligands will be their energy will be minimized. Then choose again, right click on the ligand and choose the option convert all to Autodock ligand or PDBQT format, and you will see that your ligands will be converted to PDBQT formats and are present in the ligand section of Autodock wizard. Now in the table section, you can see the toroidal angles for each ligand. Now hit the Vena wizard and you can see an option of start. Click the start button and here comes our grid box. Gridding, as I told you earlier, is the most important section of visual screening. Gridding allows us to specify a region where we aim our ligands on the receptor molecule. By using these knobs, you can just cover your ligand. For now, I'll be just moving my ligand to this section, a grid box to this section, because my ligands will have to attach to this. Okay, try to make it as constrained as possible, so my molecules this side of chain as in the grid box. Okay, I'll be just shortening it here to minimize the time limit. The larger the grid box, the more we will the pauses and the more time it will take. Once you have set your grid box, select all the ligands by holding Control key and hit forward button. This will start the docking process of visual screening. You can see it is working with each ligand one by one, and it is taking very less time. Once the visual screening docking is completed, you can see your ligand pauses here. In the load menu here, you can see the ligand, their binding affinity, mods, their RMSD values on upper bound and lower bounding values. This data can also be saved in the CSV format by clicking this icon. And for now, I am choosing this option to be pauses and save. Let's open it. And here you can see the binding affinities, the RMSD of upper bound and lower bound values. So the pose that is to be chosen should be with the least binding affinity. That means the structure is very much stable with this affinity. This can be saved in the PDB format by right clicking on this button and save as PDB. And let's just save it as lead pose. Save. And this pose can be visualized in PyMol or Discovery Studio as you prefer. Click on File button, open, and load your protein structure here. You can see, okay, we can also hide surface from here. This is our structure complete in surface format, and you can also use this visualizations further. For binding analysis or for visualizing the interaction of ligand, we will use Lig+. That's all for today. Hope you have learned a lot. If you face any difficulty or error, do let us know. Allah Hafiz.